Hello, my name is Heather MacArthur, and I'm the Clinical Director of Breast Cancer at UT Southwestern in Dallas, Texas. And it was my privilege to present at the recent ESMO Congress in Barcelona, Spain, the results of the CAPItello 290 Phase 3 study. That was a study interrogating capivasertib with paclitaxel as first-line treatment for metastatic triple-negative breast cancer. By way of background, capivasertib is an oral potent and selective inhibitor of all three AKT isoforms. It's currently recommended in combination with fulvestrant as a treatment option in ER-positive, HER2-negative advanced disease for patients who have one or more of the following mutations: PIK3CA, AKT, or PTEN, following progression on one or more prior lines of endocrine therapy. The PI3K AKT signaling pathway is often overactivated in cancer, promoting breast cancer cell survival and resistance to chemotherapy or endocrine therapy. Notably, about 1/3 of patients with triple-negative breast cancer harbor a mutation in PIK3CA, AKT, and or PTEN. Preclinical data, which was also presented at the ESMO 2024 meeting, demonstrated that combining capivasertib with paclitaxel decreased cell proliferation and increased cell death in triple-negative breast cancer cells with and without these alterations of interest. Moreover, the randomized double-blind Phase 2 LOTUS and PACT studies provided rationale for a Phase 3 trial of the combination in metastatic triple-negative breast cancer. Specifically, in both trials, progression-free survival and overall survival favored patients treated with the AKT inhibitor with paclitaxel when compared with placebo with paclitaxel, and there was a potential for overall survival noted in patients with or without one of these three tumor alterations. So here we presented the results of the Phase 3 randomized CAPItello 290 study examining the efficacy and safety of the addition of capivasertib to paclitaxel as first-line treatment in patients with metastatic breast cancer. This was a Phase 3 randomized double-blind placebo-controlled study in which 812 patients were randomized 1-to-1 to receive paclitaxel with either capivasertib or placebo. Men and women were eligible to participate if they were eligible for taxane monotherapy. They could not have received prior curative intent chemotherapy within the preceding 6 months or 12 months for taxanes. They could not have received prior systemic therapy for inoperable, locally advanced, or metastatic disease. They had to have good functional status. Because of concern about impact on glycemic levels, patients with hemoglobin A1C less than 8% and diabetes not requiring insulin were allowed to participate, and FFPE tumor samples had to be made available for central analysis. There were two primary endpoints for this study: overall survival in the overall population, and the second endpoint being overall survival in the PIK3CA1, AKT1, PTEN altered population. By way of baseline characteristics, the median age was 54. All participants were female, with about 2/3 of those being postmenopausal. The median body mass index was 26. Approximately 5% of patients were enrolled in the US and China, so the majority of patients were enrolled in the rest of the world. Over 2/3 of patients had visceral disease. Approximately 40% of patients had de novo metastatic disease. About half the patients had prior curative intent chemotherapy. Approximately 15% of patients had PD-L1 positive tumors, although notably PD-L1 status was not available for approximately 40% of the patients. And a question that we often get is this a patient population that was previously treated with immune therapy in the curative intent setting? And the short answer is no, and that only 4 of the participating patients received prior immune therapy. Alteration of frequency was consistent with the literature and balanced across treatment arms, with approximately 30% of patients having any alteration, with most of those alterations being in PIK3CA and or PTEN. And in terms of the dual primary endpoints, again, overall survival in the overall population and in patients with PIK3CA, AKT1, or PTEN altered tumors, there was no improvement in overall survival in either group. group. The overall survival result in the overall population was not attributable to any specific subgroup in interrogating all of the subgroups investigated in the forest plot provided. In one of the key secondary endpoints, progression-free survival did numerically favor capivasertib with paclitaxel over placebo with paclitaxel with a hazard ratio of 0.72 in the overall population, translating to a 0.5-month improvement in PFS. In the altered population, the hazard ratio was 0.70 with a just under 2-month improvement in PFS in that group. And the PFS benefit was consistent across all subgroups interrogated. Overall response rates also numerically favored the combination of capivasertib with paclitaxel over placebo with paclitaxel, and that the overall response rate in the overall population was 50.1% versus 37.6%. and very consistent in the altered population. The frequency of adverse events leading to discontinuation was low across all groups, although there was a higher rate of adverse event leading to dose interruption or dose reduction in the capivasertib treated population. Diarrhea was the most frequent adverse event at 76.4% in the capivasertib treated group, and it was the most frequent grade 3 or higher adverse event in the capivasertib treated group at 12.7%. However, the adverse event profile was broadly consistent with the known profiles of the agent. In honing in on the hyperglycemia, the rate of hyperglycemia was higher in the capivasertib group at 22.1%, but was mostly low grade. So in conclusion, CAPItello 290 was conducted in response to the positive data from the phase 2 LOTUS and PACT trial. The dual primary endpoints of overall survival in the overall population and in patients with PIK3CA, AKT1, and or PTEN altered tumors failed to meet the pre-specified boundary for statistical significance. In pre-specified analyses, differences were noted in the secondary endpoints of PFS and overall response rate, and numerically favored capivasertib with paclitaxel over the placebo paclitaxel population, both in the overall population and in patients with altered tumors. The safety profile of the combination was broadly consistent with the known profiles of capivasertib and paclitaxel with no new signals identified. So in summary, CAPItello 290 did not meet the predefined threshold for improving overall survival in either the overall population or in patients with PIK3CA, AKT1, and or PTEN altered tumors, although PFS numerically favored first-line capivasertib with paclitaxel over placebo with paclitaxel in patients with metastatic triple-negative breast cancer. So the key takeaways are that this was a negative study. The sponsor has reported that they will not further pursue this asset in triple-negative breast cancer. However, this data will not impact ongoing studies investigating this asset in other disease settings, such as the CAPItello 292 study, which is looking at this asset in combination with hormone therapy for hormone receptor positive disease.